Welcome, my name is Ben Westhead, and today I'll be discussing a research article that investigated the incipient resistance of Helicobacter punctidra to the cry2AB protein found in Monsanto's Bollgard 2 transgenic cotton. In this article published in 2010, it states that the three researchers involved measured the frequency level of resistant alleles to the toxin in the main cotton growing regions of Australia. This research was valuable in understanding that the pyramiding of dual toxins is not foolproof and justifies the work of the monitoring for resistance within the wild. The researchers' aim was to measure the level of resistance to the toxin cry2AB in the Helicobacter punctidra population of the cotton growing regions in Australia. Within this research, there were two main methods of measuring the resistance frequency. The first is an F1 screen, where a field moth reared from eagle larvae is mated with a moth of known resistance that has been raised from a resistant laboratory strain HP-14 and measures only two alleles. The pairs mating results in a bioassay that shows the field moth's allele configuration, i.e. if the moth was fully susceptible, the offspring produced are all heterozygote and are all affected by the protein. If the field moth was already heterozygote, 50% of the offspring produced should be homozygous resistant and 50% heterozygotes, etc. Within the F2 screen, the method is the same, only two unknown field moths are mated together and the offspring are reared, and then their offspring are tested for resistance to cry2AB and measures 4 alleles. In the research, F1 screens were performed on both populations found in cotton cropping and non-cotton cropping areas, whereas F2 screens were performed on collections from the main cotton growing regions. The results shown in the research article were from the F2 screens performed on live specimens from cropping regions and measured an increase in resistant frequency across the cotton seasons from 2004 through to 2009 of approximately 0.002 to 0.012 difference. These results prove the resistance is inevitable regardless of the introduction of dual biotechnology measures. The data from F1, which are not as complex as F2 screens, was used in comparison to the F2 data to provide a distinguishable difference in frequency of resistance between cropping and non-cropping regions. It was found that there is an about 8-fold increase of resistance frequency between the two regions. Within the article, the discussion explains that there are now two forms of evolutionary field resistance to dual toxin cotton. Firstly being the significantly greater number of resistant alleles in cropping regions in comparison of those non-cropping regions, and secondly the gradual increase of resistant alleles found in cropping regions across the 5 seasons measured. The discussion also points out the importance of a third agent to be used in the fight of pest species found in Australian cotton. This third agent needs to be specifically targeted to the 2AB resistant larvae that have survived towards the end of the season, and that this agent may benefit from not being found in the cry class of proteins. Thank you for listening.